Hello students, hope you all are doing well. In today's video, we will cover the subject hematology. The topic of the video is routine hematological test. So, in today's video, we will focus on what are routine hematological tests and what all test comes under the routine hematology. In today's video, we will let our focus on hemoglobin examination performed in the lab. What are the different methods through which we can perform the analysis of hemoglobin in the laboratory? So, without delaying, let's start the video. As the name suggests, routine hematological tests are the tests which are performed in the hematology laboratory, and these tests are performed with the help of blood sample. As you all know, you all are aware that hematology is the study of blood. So, the hematological tests are the tests which are performed for the analysis of blood samples, that which testings we can perform to detect disease in the blood. The hematological tests are performed for analyzing the infection, anemia or blood clotting disorder in the blood. There are different arrays that can be performed with the blood sample. So, routine hematological tests are the common type of laboratory tests that are performed for the analysis of blood samples. In the blood sample, we detect different diseases that may affect the blood composition or the regulation of the blood. Alright, so hematological tests are performed for detection of different abnormalities in the blood sample. Moving on. In today's video, we will lay our focus on the different testing performed in the routine hematological test. In today's video, we will focus on hemoglobin, that how hemoglobin is determined in the laboratory. Alright, so let's begin. You must all be aware that what is actually hemoglobin. Let's just first discuss a little about hemoglobin, that what is actually hemoglobin. So, hemoglobin is a conjugated protein present in the blood. The color of the blood is red due to the presence of hemoglobin. It is also known as chromoprotein because it consists of a colored pigment. Alright, so chromo means color and protein is a protein part which is made up of amino acids. So, hemoglobin consists of chromo, the colored part, which is iron. Due to the presence of iron, the color of hemoglobin is red. So, in today's video, we will lay our focus on the determination of hemoglobin concentration, that how we can analyze hemoglobin in the blood sample, the concentration of hemoglobin in the blood sample. It is very important to find out, to rule out the concentration of hemoglobin in the blood, because the examination of hemoglobin will tell us about the different hematological disorders that can be present in our blood. It can rule out anemia, if any person is suffering from anemia or not. It can also rule out what type of anemia is a person suffering from, and various disorders, hematological disorders that are related to blood can be ruled out by the determination of hemoglobin concentration in the blood. So, hemoglobin is determined in the blood by two main methods, although there are four main methods, but in today's video we will discuss about the two main methods that are commonly performed in the laboratory. The other two are not commonly performed in the laboratory. The first one is the first method that is performed for hemoglobin concentration is the Saheli's method, and the second method is the Sinamith hemoglobin method. Alright, so first one is the Saheli's method and the second one is the Sinamith hemoglobin method. We will discuss these two methods in today's video because these are the most commonly performed method in the laboratory for the determination of hemoglobin concentration. The other two methods are that are not commonly used in the laboratory are the first one is the specific gravity method that is performed with the copper sulfate solution, and the second one is the gasometric method, which is also called as indirect method for the determination of hemoglobin concentration. So these two methods are not commonly used in the laboratory. We will discuss in today's video the most commonly used method for the determination of hemoglobin concentration. So let's focus on the first method. The first method is the Saheli's method. So in Saheli's method, the determination of hemoglobin concentration is determined using an instrument called Saheli's hemoglobinometer. So what is actually Saheli's hemoglobinometer? Saheli's hemoglobinometer is a manual. So Saheli's hemoglobinometer is a manual set of instrument used for the determination of hemoglobin. Alright, what it includes, let's just have a look. The Saheli's hemoglobinometer includes a pipette, hemoglobin tube, stirrer and a comparator. So there is a box type instrument which is called as Saheli's hemoglobinometer. It consists of 3 blocks. The 2 are the glass blocks and this middle block is for keeping the hemoglobin tube. A glass tube is there in which we take our blood sample and the reagent. Then after taking the blood sample, we put the this glass tube here. This box is called as comparator box. This is hemoglobin tube, and a stirrer rod, glass rod, is used for stirring the samples in this glass tube. So this is the Saheli's hemoglobinometer. It is known as Saheli's method because it was invented by a scientist Saheli. So this is a manual method of determination of hemoglobin concentration. This method can be performed anywhere but in the presence of sunlight or ample light. Alright, so what is the principle of this method? Let's focus on the principle, that how it works, how the hemoglobin concentration is measured by this method. So what happens in the Saheli's method? We take a reagent called N by 10 HCl. What is N by 10? This is the normality of this HCl solution. So we take 0.1 normal HCl. Now what this 0.1 and HCl perform? When the blood is mixed with N by 10 HCl, the N by 10 HCl converts the blood, the hemoglobin present in the blood, into acid hematin. This is the principle. What does N by 10 HCl do? When we mix the blood with N by 10 HCl, the hemoglobin present in the blood gets converted into acid hematin by the action of N by 10 HCl. This acid hematin is a brown colored compound. It is a brown colored compound. Now what we do, when we get the brown color compound in this glass tube after mixing the blood with N by 10 HCl, we then put this tube into the comparator box and compare with the brown glass tubes that are already present in this comparator box. We compare the color of our test sample with this brown glass comparator and measure the amount of hemoglobin present in the blood sample. Let's focus one more time on the principle. What is the principle of Saheli's method? When we mix the blood with N by 10 HCl, the hemoglobin present in the blood gets converted into acid hematin, which is a brown color compound. The color intensity of this brown colored compound acid hematin is then compared with the standard brown glass which is present in the comparator box. This is the principle of Saheli's method. Now just take a look at the Saheli's hemoglobinometer. As I told you, this is the Saheli's hemoglobinometer and this is the whole kit. Alright, so here you can see this is the brown glass comparator. This is the glass tube. As you can see, this is the glass tube, also known as hemoglobin tube. Here you can see a glass stirring rod for stirring the liquid. This is the hemoglobin pipette. It does not have any kind of bulb. This pipette has one mark indicating 20 cubic per mm. So you have to take 20 cubic per mm blood sample and mix it with N by 10 HCl. This is the hemoglobin tube, hemoglobin glass tube. It is graduated on both sides. It has marks on both sides. On one side of the this is the glass tube, alright. So on one side of the glass tube, the marks are there. The marks are in gram percentage, and on the other side also it is marked. On the other side, the marks are given in gram per deciliter. Alright, so here it will be marked as 2, 4, 6, 10 and on this side it will be marked as 20, 40, 60, 80 respectively. Alright, so if we are giving the analysis of hemoglobin as gram percentage, we have to take the measurement from this side, and if we are giving the analysis in gram per deciliter, we have to measure the we have to give the analysis by this side. So this is hemoglobin tube. Next one is the comparator. You can see here, this is the comparator box. It has two glass comparator having brown color, and the middle area is left for inserting the hemoglobin tube. In this area we insert the hemoglobin tube. Alright, so this is how a Saheli's hemoglobinometer looks like. Reagents for Saheli's hemoglobinometer, or to assay hemoglobin from Saheli's hemoglobinometer, we need a reagent called N by 10 HCl. We have to make it in 0.1 normality. So to make 500 ml of N by 10 HCl, we need 4.5 ml of concentrated HCl and mix it with 500 ml distilled water. So this is how we can make normal N/10 HCl. Moving on. Now talking about the procedure, that how we can perform the hemoglobin estimation by Saheli's method. So what we have to do, we have to add N/10 HCl to the hemoglobinometer tube up to its lowest mark. As I told you, this is the hemoglobin tube. It has markings. The lowest mark is 2 and here it is 20. So up to 2 mark we have to put N/10 HCl in this tube. Alright. After adding N/10 HCl, we have to mix blood sample. We have to take the blood sample. EDTA blood sample is used for performing the hemoglobin estimation. Alright. So in hemoglobin pipette up to its 20 cubic mm mark, we have to take the blood sample, EDTA blood sample, and then we have to mix this blood sample into this N/10 HCl in this hemoglobin tube. Then we have to leave the solution for 10 minutes. After stirring with glass rod, we have to leave this sample in which we have mixed N/10 HCl with blood. We have to leave it as it is for 10 minutes at room temperature so that hemoglobin can be converted into acid hematine. After 10 minutes, we have to add distilled water drop by drop and mix by the stirrer until the color in this tube matches with the color of the brown glass comparator present on the comparator box. Alright, what we have to do, there is a brown glass comparator, right? Here we have to put the hemoglobin tube and at both sides it has brown glass comparator. So when we leave the solution for 10 minutes, then after the 10 minutes we have to insert the hemoglobin tube here. Now what we have to do, we have to add distilled water drop by drop to this tube so as to match the color of the solution with the brown glass comparator present on both sides of this comparator box. For mixing, each time we add distilled water, we have to stir the solution with the help of stirrer, glass stirrer present in the Saheli's hemoglobinometer kit. When the color of the solution matches with the brown glass comparator, we will stop adding the distilled water, and for the estimation of hemoglobin, we have to put out this hemoglobinometer tube and keep it at the level of the eye, and for taking the measurement, we have to look at the lower meniscus of this solution in this hemoglobin tube, and the readings at the lower meniscus of the glass tube represents the amount of hemoglobin present in the blood sample. For example, if the readings here are 12, we have to assess that the blood sample consists of 12 gram per deciliter hemoglobin concentration. Alright, so this is the procedure of hemoglobin determination by Saheli's method. Let's have a look at the advantages and disadvantages of this method. The advantages, first of all, it is inexpensive. It does not include the high cost reagents or high cost technique, so it is inexpensive. Second one is it is easy to perform. It does not have a complex procedure. As well as there is one more advantage, that you can perform it anywhere. The kit is easy to handle and take, so you can take the kit with you where you have to perform this test. Alright, now look at the disadvantage. Acid hematin is unstable, so the color fades away quickly. So it is time bound. If you are leaving the solution for 10 minutes, you have to keep a check that the time is 10 minutes only. It should not exceed the 10 minutes incubation period. Second is visual error. Visual error is possible while matching the brown color of the comparator box, because for one it may seem that the color has reached, for the other person it may seem that the color has not matched with the brown glass comparator. Then technical errors may be present, like improper mixing of blood or error in pipetting and capillary blood with tissue fluid. So these errors may be involved while performing this technique. Nowadays in laboratory, manual method is not generally used. In laboratories which does not have modern equipments, only perform hemoglobin determination by Sehli's method. Moving on. Next method for determination of hemoglobin is Cynometh hemoglobin method. This is more reliable method for the hemoglobin determination. The labs generally use this method for the determination of hemoglobin. Alright, so Cynometh hemoglobin method is also called as, let's just have a look at the names. It is also known as colorimetric method, because it includes the assay by instrument called colorimeter. So by the help of instrument colorimeter, we measure the intensity of hemoglobin in this method. Now what is the principle of this method? Let's have a look. In this method is also known as Drabkin's method. You should always know the different names, because the question may arise by different name also, that explain Drabkin's method. So you should always know that what are the other names of any method or any thing. Alright, so now we will focus on this Cynometh hemoglobin method. It is also known as Drabkin's method because it includes the determination of hemoglobin by the reagent called Drabkin's reagent. In this method, we use the reagent called Drabkin's reagent. So let's focus on the principle of this method. Let me remove this. Let's have a look now. When blood is mixed with Drabkin's reagent, there is a reagent called Drabkin's which we use in this method. When the blood is mixed with Drabkin's reagent, the potassium ferricyanide present in the Drabkin's reagent converts the hemoglobin present in the blood into methemoglobin. A methane group is attached with the hemoglobin and converts the hemoglobin into the form methemoglobin. Then, after the conversion of hemoglobin into methemoglobin, potassium cyanide present in the Drabkin's reagent reacts quickly with the methemoglobin and converts this methemoglobin into Cynometh hemoglobin, which is a light pink colored component. The intensity of this colored solution is then measured in the colorimeter. This is the principle of Cynamethyl hemoglobin method. When the blood reacts with or is mixed with Drapkin's reagent, the potassium ferricyanide present in Drapkin's reagent converts the hemoglobin into methemoglobin. The potassium cyanide present in the Drapkin's reagent reacts quickly with the methemoglobin and converts it into Cynamethyl hemoglobin, which is a light pink colored compound. The intensity of this light pink colored compound is then measured in an instrument called colorimeter. Now, how the colorimeter measures the intensity of color solution? The colorimeter is an instrument which works on Beer-Lambert's law. The colorimeter instrument works on the principle of Beer-Lambert's law. According to Beer-Lambert's law, the intensity of color solution is directly proportional to the amount of solute present in that solution. According to Beer-Lambert's law, the intensity of colored solution is directly proportional to the concentration of solute present in any solution. So we can say the darker the color, more the intensity. Darker color, more intensity, and lighter color, less intensity or less concentration of the solute. The intensity of hemoglobin is measured at 540 nanometer using green filter in the colorimeter. Hope this principle of Cynamethyl hemoglobin method is now clear to you all. That when the blood is mixed with Drapkin's reagent, the potassium ferricyanide present in the Drapkin's reagent converts the hemoglobin into methemoglobin. The potassium cyanide present in the Drapkin's reagent again reacts quickly with the methemoglobin and converts this methemoglobin into Cynamethyl hemoglobin, which is light pink colored compound. The intensity of this light pink colored compound is then compared at 540 nanometer in a colorimeter. Moving on. So this is Drapkin's reagent. It is commercially available. For assaying the hemoglobin determination by colorimetric method or by this Cynamethyl hemoglobin method, we need an instrument called colorimeter, we need Drapkin's reagent and a standard solution. Standard solution is used for comparing the values of the test. Alright, so how we can make Drapkin's reagent? We need potassium ferricyanide 200 mg, potassium dihydrogen phosphate 140 mg, potassium cyanide 50 mg, non-ionic detergent 1 ml, and add all these compounds to distilled water and make the volume up to 100 ml. The pH of the Drapkin solution must be 7.0 to 7.4. This is the composition of Drapkin's reagent. Moving on. Now focusing on the procedure of this Cynamethyl hemoglobin method. To perform Cynamethyl hemoglobin method, we need 3 glass test tubes and we have to mention it as blank, standard and test. Alright, we need 3 test tubes and label it as blank, standard and test. Now we have to add 5 ml Drapkin solution, or we can add 5 ml we need to add, we have to take 3 test tube and label it as blank, standard and test. In blank tube we have to add 5 ml distilled water. In standard solution we need to add 5 ml distilled water, and in test solution we need to add 5 ml Drapkin's reagent. In the standard solution we have to take 5 ml Drapkin's reagent, and in test solution we have to take 5 ml Drapkin's reagent. Now what we have to do, we have to maintain the volume in all the 3 test tubes. So here we will add 20 microliter buffer with 5 ml distilled water. In a standard tube we will add 20 microliter standard solution, and in test tube we have to add 20 microliter of blood sample. So the volume is maintained. Now we have to leave it for 5 minutes at room temperature for incubation, so that the reaction can occur easily in all the tubes. Now after 5 minutes we have to measure the intensity of these tubes in the colorimeter. First of all, in colorimeter we will put the blank tube and take the reading and keep it as 0. There is a 0 tab in the colorimeter, so blank is used to adjust the reading to 0, so that no error could be there in taking the readings of standard and test solution. So first of all we will take the reading of blank solution, we will make it to 0. Then we will put it out and insert the standard solution tube here. We will put the standard tube here, and in a standard tube the colorimeter will now give the reading of standard solution. The reading will appear here. It is mentioned as OD or absorbance. We will take the reading of standard solution and then we will put out the standard tube. After this we will take the reading of test solution, which is here, in which we have mixed our blood sample. Now test tube will be there and it will give the OD of test or absorbance of test. We will note the OD of standard and test. After noting down the ODs of standard and test, we will put it in the formula. Here is a formula. 15, which is the concentration of standard, multiplied by OD of test upon OD of standard solution. So with this formula we will get the concentration of hemoglobin in gram per deciliter. This is how we will evaluate the hemoglobin determination by Cynometh hemoglobin method. Let's have a look at the advantage and disadvantage. There is no visual error, so it is more reliable method. Cynometh hemoglobin is stable, so it does not fade away quickly. You can get the results. All forms of hemoglobin except sulfohemoglobins are converted, so it gives a proper or accurate determination of hemoglobin. This is a reliable standard reference directed by WHO, so it is used most commonly in the lab. Now what are the disadvantages? It contains potassium cyanide, which is poisonous, so it should never be pipetted with mouth. After dilution, solution should be kept for some time for complete conversion. You cannot get it rapidly, so you have to wait for 5 minutes. This is a disadvantage. Rate of conversion of blood containing carboxyhemoglobin is long, and blood with abnormal plasma proteins and high leukocyte count may cause turbidity on dilution of blood. So it can give an error while assaying in the colorimeter. So these are the methods of determination of hemoglobin. Hope you enjoyed learning. Keep connected and keep studying. Thank you for listening.